Look, the first step of protein synthesis called transcription. We said the process consists of two steps, right? Transcription and translation. Transcription and translation. Now we illustrate the transcription process. First, you have to know that what occurs. لتشين برضو ذاتا. Transcription in the eukaryotic and also in the prokaryotic. In the eukaryotic, we have a nucleus envelope which separates the DNA from the cytosol. So in the eukaryotic, it occurs in the nucleus. But in the prokaryotics, which lacks the nuclear envelope, which separates the DNA from the cytosol, the process occurs in the cytoplasm. First one. Second one, you have to know that the transcription meaning what? Transcription, it means that the genetic information on a gene. For example, we have a gene on DNA. The information is transcripted, copied, rewritten into the RNA. It means that DNA acts as a template for what? For making an RNA. Also, we said that a gene, right? First one. Second one, you have to know that the process occurs in 3 steps, but first look at the figure. In the first step, this one, the DNA is this one. DNA is a double-stranded nucleic acid. Also, we have a gene which has promoter and it has also terminal signal. First, we have an enzyme called RNA polymerase. RNA polymerase, it do what? It binds to the promoter. After binding, the 2 strands separate. Okay? After separating, the process begins. Then RNA polymerase adds complementary nucleotides to 1 strand only. Look, in the first step, we have an enzyme called what? RNA polymerase. Binds to promoter. After binding, the 2 strands do what? The 2 strands separate, but be careful, until the termination signal, not completely. It means that not completely the both strands separate, only until the termination signal. So what about the promoter? Promoter are nucleotides on DNA. First. 2. RNA polymerase binds to the promoter. RNA polymerase binds to it. 3. After binding, you have to know 2 things about what happens after binding. Transcription begins, starts, initiates, and also the 2 strands separates. It means that the 2 strands separate after the RNA polymerase binds to the promoter. This is the first step. Also be careful, helicase is not needed in this process. Then, in the second step, RNA polymerase, it do what? It adds RNA complementary nucleotides. It uses only 1 strand as template. It adds RNA complementary nucleotides. RNA complementary nucleotides, we have PS, G, P, S, U, P, S, C, and also we have PS, A. RNA polymerase adds RNA complementary nucleotides only to 1 strand, not both, because RNA is a single-strand nucleic acid. So when there's T in the DNA, it adds what? A. Also, when there is G, it adds C, and so on. A, U, C, G, T, A, until it reaches what? Until it reaches the terminal signal. But be careful. We have a question. Which from the following determines the sequence of nitrogenous bases, you can say nucleotides in the mRNA or in the RNA? Of course, you have to know that the complementary base pairing. It means what complementary base pairing? it means that T only pairs with A, and also A with U, and also C with G. So complementary base pairing determine the sequence of nitrogenous bases and sequence of the nucleotides in the mRNA. First one. Two, also you have to know some other things about the process. In the second step, first, RNA polymerase adds free RNA nucleotides only to one strand. Be careful, not both. Two, during DNA replication and also during transcription, the complementary base pairing determine the nucleotide sequence in the newly made RNA. For example, if you have A, T, C, G, A, C in the DNA, the RNA polymerase adds U, A, G, C, U, G. That's all. Okay? Three, unlike DNA replication, during DNA replication, the DNA polymerase, it uses only a gene. For example, during DNA replication, the DNA polymerase, it uses both strands as a template, but be careful, during translation, RNA polymerase, only it uses a specific region. It means only a gene on DNA as a template, first one. Two, after the RNA polymerase moves past on this gene, what happens? The two DNA strands rewind, okay? Out, second step. Third step, when the RNA polymerase reaches the terminus signal a specific nucleotides on DNA. After the RNA polymerase reaches the signal, this stopping signal, what happens? It releases both. Look, it releases both. According to the figure, after it reaches the terminus signal, it releases both DNA and the newly made RNA. And then the two DNA strands rewind. Out. And this RNA that formed finally, it will be rRNA or mRNA or tRNA. And also you have to know some other things about the process. DNA polymerase, during DNA polymerase, and DNA polymerase, it adds A to T during replication, but RNA polymerase, it adds A to T also during what? During transcription. This means what? For example, if we have DNA as a template, this is DNA, if you have A, G, C, T, during replication, DNA polymerase, it adds what to A, T, and for G, C, for C, we have G, and for T, we have A. But during transcription, also we have DNA, but this RNA was formed, for example, we have A, G, C, T, it adds U to A, right? But this one will be the RNA. And also A to T, and also you know the other ones. That's all.